Hi, myself Sweshta Mandal from class 9 section B. Today I'm going to introduce the biology magazine topic, that is genetic engineering. Genetic engineering. The possibility for recombinant DNA technology emerged with the discovery of restrictions enzymes in 1968 by Swiss microbiologist Werner Auber. The following year, American microbiologist Hamilton O. Smith purified so-called type 2 restriction enzymes, which were found to be essential to genetic engineering for their ability to cleave a specific site within the DNA, as opposed to type 1 restrictions enzymes which cleave DNA at random sites. Drawing on Smith's work, American molecular biologist Daniel Nathans helped advance the technology of DNA recombination in 1970 to 1971 and demonstrated that type 2 enzymes could be useful in genetic studies. Genetics engineering based on recombination was pioneered in 1973 by American biochemists Stanley N. Cohen and Herbert W. Boyer, who were among the first to cut DNA into fragments, rejoin different fragments, and insert the new genes into E. coli bacteria, which then reproduced. Genetic engineering, the artificial manipulation, modification, and recombination of DNA or other nucleic acid molecules in order to modify an organism or population of organisms. The term genetic engineering is generally used to refer to methods of recombinant DNA technology which emerged from basic research in microbial genetics. The techniques employed in genetic engineering have led to the production of medically important products, including human insulin, growth hormone, and hepatitis B vaccine, as well as the development of genetically modified organisms such as disease-resistant plants. I am so honored to be a part of this biology magazine and also glad because I got a chance to introduce the topic. Thank you so much to all the teachers along with the principal and the vice principal of my school.
Formation of insulin by bacteria. Hello all, I'm Sanchita Pati from 10C and I'll be telling about how human insulin can be produced with gene editing. Genetic engineering helps us in various ways in the modern world. One such use is production of human insulin by a bacteria. It sounds quite astonishing and weird that human hormones can be prepared inside a bacterial cell. It is indeed a very difficult task. Eli Lilly was the first one to do this job. In this process, firstly, the human gene producing insulin is isolated. A plasmid from bacterial cell is also isolated. A special restriction enzyme named restriction endonuclease is used for cutting a segment of the circular DNA of bacteria where the human gene will be incorporated. Now, the human gene is attached by DNA ligase to the bacterial DNA in which the part was cut off. Now, the DNA in which the foreign gene was attached came to be known as rDNA or recombinant DNA. Finally, the plasmid or the rDNA is inserted back into the bacterial cell, and now the bacteria is able to produce human insulin by transcription and translation process. Generally, bacteria like E. coli or Saccharomyces cerevisiae is used for such process. Such organisms in which gene editing is done are known as GMO, genetically modified organism, or transgenic organism. This is how insulin is produced by bacterial cell. Thank you.
modified pest-resistant cotton variety. Strains of the bacterium Bacillus thuringiensis produce over 200 Bt toxins, each harmful to different insects. Most notably, Bt toxins are insecticidal to the larvae of moths and butterflies, beetles, cotton bollworms, flies, and insects, but are harmless to other forms of life. The gene coding for Bt toxin has been inserted into cotton as a transgene, causing it to produce their natural insecticide in its tissues. In many regions, the main pests in commercial cotton are the Lepidopteran larvae, which are killed by the Bt proteins in the genetically modified cotton they eat. Bt toxin was created through the addition of genes encoding toxin crystals in the cry group of endotoxin. When insects attack and eat the cotton plant, the cry toxins or crystal proteins are dissolved due to the high pH level of the insect's stomach. The dissolved and activated cry molecules bond to cadherin-like proteins on the cells of the brush border of epithelium in the insect's gut. The important advantages of Bt cotton are that they increase yield of cotton due to effective control of three main types of bollworms, that is, the American bollworm, the spotted bollworm, and the pink bollworms. Insects belonging to Lepidoptera are sensitive to a crystalline endotoxin protein produced by the Bt gene, which in turn protects the cotton from bollworms and leads to potential reduction in the cost of cultivation. In India, Bt cotton is mainly supplied in Maharashtra by the agri-biotechnological company Maheko.
Hello, I am Shivaditya Mukherjee from class 9B. So today we are going to discuss about hemophilia and gene therapy. Hemophilia is a rare genetic disorder which occurs due to prevent of clotting of blood properly, which may result a prolonged external bleeding. Internal or external bleeding may be very dangerous for the people with hemophilia, as it can lead them to arthritis and also some life-threatening issues. Arthritis. Arthritis is an inflammation of two or more joints causing pain and stiffness that can worsen with age. Hemophilia occurs due to lack of genes responsible for clotting. These are called clotting factors. There are 12 clotting factors. A person having hemophilia A, that means he or she lacks clotting factor 8. A person hemophilia B, that means he or she lacks clotting factor 9. Gene therapy. The introduction of normal genes into cells in place of defective ones in order to correct genetic materials. A functional version of the missing gene is delivered to the cell so that they can produce the missing factor. It is delivered by using a vector. These are actually derived from the viruses. All viral genes are removed before delivering into the cell, as it would be targeted to the liver. As liver produce all clotting factors and these vectors are being targeted into the liver, so the liver can able to produce the missing clotting factors. Thank you. That's all for today.
Good day, everyone. I, Ninisha Henrietta Daniel of Class 9C, will be discussing the drawbacks and limitations of gene editing. Gene editing, a technology that allows scientists to modify an organism's DNA, has the potential to transform many areas of science and medicine. However, like any new technology, there are also several potential disadvantages, first of which are safety concerns. One of the primary concerns with gene editing is that it could have unintended consequences. For example, making a change to one gene could inadvertently affect other genes or disrupt pathways, potentially leading to harmful effects. Gene editing could also potentially exacerbate existing inequalities in healthcare, as those with more resources and technology would be able to benefit from it, while others would not. It is also a relatively new technology, and the long term effects of gene editing on humans and the environment are not yet fully understood. Overall, while gene editing has the potential to revolutionize medicine and science, it is important to proceed with caution and carefully consider the potential risks and benefits from it. Thank you.
Hello everyone, I am Atmadeep Mukherjee of Class 9B, and today I, on behalf of my team, would like to thank God for giving us this opportunity to discuss about the revolution of the future, gene editing. We would also like to thank the Managing Trustee, Mrs. S. E. Broughton, our Chairman, Mr. J. G. Broughton, for their continuous support. We would also like to thank our Principal Sir, Mr. Bobby Baxter, our Vice Principal, Mr. Shantan Mukherjee, our Teacher in Charge, Mr. Lana Jennings, and the Junior School Coordinator, Mr. Cheryl and Day. We would also like to thank all the teachers and students who made this virtual discussion possible. Last but not the least, we would also like to thank all of you, our viewers. You are a great support to us. Thank you.